Welcome to Lab 3. In this lab, we're going to be covering inheritance, looking at how our traits are passed on from one generation to the next, and some complications in that sense. The first thing we're going to do before we talk about inheritance and alleles and genes is we're going to talk about Gregor Mendel. And so Gregor Mendel is known as the father of genetics. He crossbred pea plants in the monastery garden, and he tracked the expression of different characteristics. These characteristics being flower colors, stem length, pea color, pea shape, and so on. And he tracked these expressions across multiple generations. He identified two key principles after his tracking was analyzed, which are the law of segregation and the law of independent assortment. So the first one is the law of segregation. And so at the time, many assumed that traits from parents were blended in their offspring. So what this meant was that if a tall pea plant and a short pea plant were bred, we would then expect their offspring to have an intermediate height, right? This was the thought, this was the assumption at the time. Mendel recognized that this was not the case, and he recognized that most of the offspring pea plants were tall. So he then interpreted this to mean that the traits are controlled by distinct units, and these units occur in pairs with one inherited from the mother and one from the father. So this meant that the units for the traits or genes appeared separately in the sex cells of the parents and are brought together in the offspring. The units are segregated and then they're reunited. The law of segregation explains why genes from two homologous chromosomes are equally likely then to be passed on. Next is the law of independent assortment. Mendel noticed that the pea plants were often inherited separately. The traits in the pea plants were often inherited separately from one another. So an example would be not all of the tall pea plants had yellow seeds. So this meant that the units for these traits were sorting independently of one another in the offspring. So we know that this happens during meiosis, during that crossing over period. And so when gametes are formed, the alleles for one trait segregate into the daughter cells independently of the alleles for another trait. This is particularly the case with genes found in different chromosomes. In contrast, when genes are located on the same chromosomes, they are more likely to be inherited together and serve then as an exception to the law of independent assortment. In the module, I have added extra videos explaining the law of independent assortment and the law of segregation a little bit better and in a little more detail. I highly suggest taking a look at those videos so that you can really understand these two laws and how they work. All right, so here's a look at the pea plants, right, and the traits he was tracking. He was looking at the flower position, whether it was axial or terminal, the flower color, whether it was white or purple, plant height, tall or short, pea shape, round or wrinkled, color, yellow or green, the pod, whether it was inflated or constricted, and then the color of the pod, whether it was yellow or green. And so with this, Mendel noticed that some versions of a trait were more likely to appear in the next generation than others. And with that discovery, he discovered that some versions of the traits then were dominant and others were recessive. The dominant alleles would then mask the effects of the recessive alleles. And so with dominant and recessive, right, we're looking at genotype and phenotype. And so the actual genetic material is the genotype. The genotype is written in a way that can indicate dominant or recessive alleles present. So there are three options for genotypes. We can have homozygous dominant, homozygous recessive, or heterozygous. And then the actual expression of an organism's genetic material is what we call their phenotype. So that's the physical expression. So I think of genotype, I think of genes, and I think of the letters. I think of phenotype, I think of pH pheno, pH physical, so physical expression of those genes, what we actually physically see. So if you have a person's genotype, you can then infer their phenotype, right? We have the genotype. We can then take that genotype and say, okay, they are homozygous recessive, so they physically express their phenotype is the recessive gene, right? So if you have their genotype, you can infer their phenotype. However, if you have their phenotype, it's a little difficult to infer the exact genotype because a person with a dominant expression of a gene can either be homozygous dominant or heterozygous. So there are two ways to map out a person's inheritance. One way being Punnett squares and another way being pedigree diagrams. So Punnett squares they diagram the possible genotypic outcomes for the mating of two organisms. One parent's genotype is written above the boxes and the others on the left. And so we're going to look at a few Punnett squares really quick. So here's one. And here we see that parent number one here at the top is homozygous, specifically homozygous dominant. And here, parent number two is heterozygous, right? And so this is one gamete and this is the second gamete. And so we then take that down and this across and we get with the possible outcomes, right? It's important to remember that Punnett squares show us the possible outcomes and not what will happen if the parents have further children. So is this each time they produce offspring, it's the possibility of being any one of these. So here you have a homozygous dominant, so 50% chance of being homozygous dominant. And then here we have heterozygous, so 50% chance then of having a heterozygous offspring, right? For whatever trait this is. You can think of it as tallness, with the big T being tall, little t being short, right? Therefore, if we're taking this, big T tall, little t short, these individuals would be tall phenotypically. These individuals would also be tall phenotypically. So 100% phenotypic expression of tallness. However, genotypically, we have 50% homozygous dominant and 50% heterozygous. Right? Next, we're going to look at a dihybrid cross. And with the dihybrid cross, we are doing the exact same thing, except we are tracking two traits. So initially, we were tracking just a, right? And we would only look at that, and that's why we have a 2x2 square, right, for our Punnett square. Now we're looking at dihybrid crosses, and so we're looking at tracking two traits at the same time. So say we want to say parent one obviously is heterozygous for both traits, and we'll say A is for, I don't know, tallness, and B is for brown hair versus black hair. So big A is tall, little a is short, big B is brown hair, and little b is black hair, right? We're just, making up random traits. And so the way that you do this is you want to find the gametes, right? Here we had the genotype of the parent was homozygous dominant, big T, right? We split that up. And here we have, again, heterozygous, so big T, little t, and it's split up. We're going to do the same thing here, except we are tracking two traits. So the way that I think about it is I like to think of it as math. And I know that might sound a little scary, especially if you haven't taken math in a while, or it's been, you really don't like math. But I think of it as math, and I think back to beginner algebra when they taught you the FOIL it out method, right? Because that's exactly what we're doing. And it's not scary at all, I promise. It is very, very simple. Let me just take the pen and we'll see. And what I mean by FOIL it out is you are going here and then here. Sorry, my arrows are a little weird. And then you go here and then you go here. Very simple. Same thing down here is here to here, here to here, right? And then FOIL this out. I don't remember what FOIL it out means anymore, or what it means. So now we have this, right? And so let me go back to, let's do laser pointer. And so we have carried this over, we have foiled this out, big A to big B, big A to little a to big B, little a to little b. And so now we have 4 possible gametes that this parent can give for their offspring, right? Right? So your 4 possible gametes is big A, big B, big A, little b, little a, big B, and little a, little b. And that's exactly what we have with our gamete possibilities. Parent number 2 gamete possibilities then are big A, little b, big A, little b, little a, little b, and little a, little b. And that's exactly what we have here. So now that we have our 4 gamete possibilities, we have to put them into our Punnett square for a dihybrid cross. And so that then takes us then to this dihybrid cross, this monster of a cross. It looks very complicated, but again, it is very, very simple, right? We're going to plot our gametes here for parent 1, plot our gametes here for parent 2, right? And we have a 4 by 4 Punnett square. Ignore the gray boxes, right? Usually these would just be written on top of it. It's just a lot easier to do it this way. So again, our 4 gametes for parent 1 are big A, big B, big A, little b, little a, big B, little a, little b. And then for parent 2, our 4 gamete possibilities are big A, little b, big A, little b, little a, little b, and little a, little b. And so now you drop them down and you drag them across, and that's how you plot out what the offspring potentially are going to have genotypically, and then you can calculate out phenotypically. So child 1, and you can pause the video now and you can try it out on your own, plot it out and see what you get, and then double check. So I'll give you a minute, pause the video, and then you can come back. Right? So now we're going to plot it out and you can check if your answers are correct. So for the first box, this box right here, you drag this A down, this A across, this B down, this B across, and you get big A, big B, little b, right? Big B, little b, right? When we're doing dihybrid or any type of genotypic expression, if there is a dominant trait, just put it first. It doesn't matter which parent it comes from. Just do it first. It's easier to visually see it. All right? Again, you drop it down, take it across, same thing, big A, big B, little b. Easy, right? Down here, you bring this A down, this A across, this B here, this B across. So you have big A, big B, little b. That's exactly what we have here. And then the same thing, bring it down, drag across. You have big A, big B, little b. All right, we've done the first row. Next, we'll do the next row. So big A, little b, right? Right there. And then down across again, big A, little b, exactly the same. Down, big A, little b, So we have here, again, big A, big B, little b. Then you go to the next row, little a, big B, little b. But remember, we're going to keep it for clear, to make it clear, organized, right? We'll do big A, big B, little b. It doesn't really matter. It's just this is a lot cleaner to see if you have the dominant first. Then you have, again, little a, big B, little b. So we have big A, big B, little b. Down and across again, little a, big B, little b. Right? So here we go, little a, big B, little b, right? And the A's will always come first, the B's will always come first, or whatever letters you're looking at, right? Just keep it consistent, right? Little a, big B, little b, again. And then the last row is little a, little b, little b. So we have here, big A, little b, little b. And then again, it's a double, right? Big A, little b, little b, right? And then down here, the last two are little a, little b, and you're going to get the same thing for both. In this case, because this parent gives off the same 2 here and the same 2 here, this line is the same as this line is the same as this line. So now, what you're going to want to do is look at this and try and calculate or understand what we're looking at, right? So we said the big A is going to be tallness, little a is short, and we said big B is brown hair, little b is black hair. So we're going to decipher this, right? This outcome here would be tall with brown hair, right? Because one dominant allele takes over everything, right? So the same thing here. Here we have tall with black hair, right? Because we have 2 recessive alleles, so they'll come out with black hair. And then one or more dominant alleles will give you a dominant phenotypic expression, right? So same here. Big A, little a, they're going to be tall. Big B, little b, they're going to have brown hair. So here. Little a, big A, so big A, little a, right? It means they're going to be tall. And then they're going to have black hair, right? Right, so we have here. Then we have, let's see, tall and brown hair, right? We have 2 here. Tall, black hair, tall, black hair, short with brown hair, short with brown hair, and then short with black hair, short with black hair, right? And so on the quiz at the end of the week, I will ask you to make a dihybrid cross. You're going to look at the dihybrid cross. You're going to calculate out. You're going to find it out. And then I'm going to ask you percentages. I'm going to ask you, what is the possibility of a child having, in this case, being short with black hair? And so what you're going to do, and the easiest thing to do is you have 1, 2 here out of 16 total. 2 out of 16 is 1 out of 8. I don't think I will give you like percentage numbers. It might just be fractions. So look at it that way. And you'll do that. Or I'll say tall with brown hair, right? And so then you're going to go, right? You're going to get your pen. And actually, let's do a highlighter, right? Tall, brown hair. Tall, brown hair. Tall, brown hair, tall, brown hair, tall, brown hair, tall, oh, wait, no, not that one. Sorry, ignore that one. Let me see if I can get an eraser. Eraser. There we go. Let's go back. Let's do highlighter again. Tall, brown hair. And let's see, tall black hair, short black hair, short black hair. Short brown hair, short brown hair, tall black, tall black, tall brown, or no, yeah, tall black and tall black, right? So now we have 1, 2, 3, 4, 5, 6 out of 18, right? Or about 1/3 of the likeliness of having a child that is tall with brown hair, and that's exactly what you're going to do, right, on the quiz. Fairly simple. So there we go. Those are dihybrid crosses, right? So Punnett squares, super easy, right? Right. If I were to give you a Punnett square on your quiz, it'd be like giving you free points. We want to practice that we understand a more complicated version. So your quiz, you're guaranteed to get a dihybrid cross, not a regular 2x2 Punnett square, right? So the next thing we're looking at is pedigree diagrams. And so pedigree diagrams show multiple generations of real related organisms. This is different from a Punnett square because a Punnett square shows only one real generation, which is the parents, and potential generations, or the possibility for a single offspring. Pedigree diagrams also show phenotypes for a trait rather than genotypes. So before we were looking at genotypes, here we're looking at phenotypes. So genotypes, though, can be inferred from the information on the pedigree, but we cannot be 100% confident in the interference sometimes. So pedigree diagrams include several symbols to represent the real individuals. Squares are males, circles are females. Mated pairs are linked by a horizontal line between their symbols, and then offspring are then drawn as descending from their parents' mated pair. When when a shape is colored in, it means that the person expresses that trait. Uncolored means that they don't express it. You want to take note then, right? Make sure you keep this, in mind. It does not mean that either dominant or recessive traits may be shaded in the diagram. It depends on what is being expressed or what is being represented, right? So, freckles, I believe, are a dominant trait. Here we are this is the example straight from the book, right? We're doing a pedigree diagram for freckles, right? So we can look here and say we know individual B, C, and D have freckles, right? That means individuals A, E, G, and F don't have freckles, right? Because they're not shaded in. Freckles, again, is a dominant trait. So in order to not express freckles, it means they have to be homozygous recessive, right? So that means then that their genotype, right, we can automatically say is little f. And I know these look like big F's, so I'm going to see if I can curve. Oops. If I can curve this a little bit better, right? Pen, right? Right, sorry, that F looks weird. Right, little f, and it's going to be the same, right, for this individual here. Right, follow through with all of them. And then individual G as well. Right, and individual A. Right, so now we have individuals A, E, F, and G. Their genotype is secure. They are not expressing freckles, and freckles is a dominant trait. Therefore, in order for that to happen, they must have two recessive traits, which we know as homozygous recessive. Now we're looking at individuals, then the easiest way to do is look at individuals C and D. They both express freckles. So we know that they need at least one dominant gene, right? One dominant allele. But their father here is homozygous recessive. He can only give off a recessive trait, right? So I'm going to put it here. So his recessive trait here gives it to his daughter, right? And the same for the son, right? He can only give these away. So that means they can only then be heterozygous, right? Because they're expressing the trait. That means they inherit that dominant trait from their mother, right? So here's big F, right? And so we have everybody, and now we have their mother. And so their mother is a little bit more complicated. We only know based off of her children and the fact that she is shaded in, that she expresses a dominant trait. We don't know whether or not she is heterozygous or if she is homozygous dominant, right? So in this case, we would say she is homozygous dominant, or she is heterozygous, right? And that's how we fill out this this pedigree diagram. Say they had a third child, and that third child did not express freckles, meaning that third child was homozygous recessive, then we would have enough information to know that the mother is heterozygous, right? She could not be homozygous dominant, because then all her children would need to express freckles, right? But that's hypothetical, right? We're going based off of this pedigree diagram right now. So let's check, right? Let's check with the slide. So individuals A, E, G, F are homozygous recessive, C and D are heterozygous, and then B can be heterozygous or homozygous dominant. If we check, right, that's exactly what we get, right? Homozygous recessive, homozygous recessive, 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 heterozygous, and then either homozygous dominant or heterozygous, right? And that is exactly what we get. Again, we're not looking like saying, okay, shaded means dominant and unshaded means recessive. That's not what we're saying, right? We are saying shaded is expressing the trait, unshaded is not expressing. It depends on the trait that you are talking about, right? In the labs, I believe, and maybe on the quiz, I forget, I could ask for a recessive trait to be that we're expressing, and then you have to really pay attention to what I'm asking you, right? If we look at a diagram for the recessive trait of attached earlobes, individuals who have lower earlobes than that attach to the sides of their head, that is a recessive phenotype, that will be then shaded in the diagram, right? So let's say in this case, let's go back to here. Let me clear out all the annotations, right? And say we're doing attached earlobes, and I want to see, okay, we're going to say individuals, we're tracking the attached earlobe, right? Attached earlobe is a recessive trait, right? So let's do this here. We'll do E for earlobe, right? And that'll be, let's do pen. So big E, I'll do it over here. Eraser. Right, pen. So big E is going to be unattached. Right, I'm just going to shorten it there. And then little e is going to be attached. A-T-T. I'm just going to leave it like that. So now we're looking at attached earlobes. That is a trait we are tracking with this pedigree diagram. So in this case, right, we know then, okay, individuals B, C, and D are expressing attached earlobes. Therefore, they are homozygous recessive, right? Because they are expressing that recessive trait. Sorry. And then D as well. Oh, that one was terrible. There we go. And then we know that these two children must inherit a recessive trait from their father, right? So we have little e, but they are not expressing attached earlobes. They have unattached earlobes, right? So that means they are heterozygous, right? Big E, little e, right? Again, we don't have enough information for individual E or individual A, so they can be big E, little e, or big E, big E. Very hard to do this with the mouse. Big E, little e, or big E, big E, right? So that's the case, right? We're tracing here a recessive trait, okay? Yep, so that is that. So that is a pedigree diagram. Again, it is very, very simple. It's just, you're just putting a puzzle together is what you're doing. You're looking at, okay, what is the immediate information that you can get? The thing I would do first is write down automatically the genotypes for anyone who is expressing the recessive traits, right? In this case, those are our shaded in, right? In the previous example, it was our non-shaded in, right? So again, write those first. And then from there, carry it out. See what else. Okay, this individual has 2 children. He can only give off a recessive gamete, so automatically recessive gametes go down. Then we look, okay, they are not expressing this attached earlobes, therefore they must have a dominant trait, right? Big E, big E. Boom. We have our homozygous recessives, we have our heterozygous, and then you look at our homozygous dominants, and then we see what we can get from there. Okay? Very, very, very simple. It's just a puzzle. So now moving on, right? Moving on to Mendelian traits and polygenic traits. So Mendelian traits are traits that are controlled by one gene. That is what we have been looking at, right? The attached earlobes, the hitchhiker's thumb, the freckles, right? These are Mendelian traits. There may be multiple alleles for a gene, but only one genetic locus is involved. What we mean by genetic locus is the location of a gene on a chromosome. Some Mendelian traits in humans are the attachment of the lower earlobe, with unattached dominant, attached being recessive. Hitchhiker's thumb, so not having a hitchhiker's thumb is dominant, having a hitchhiker's thumb is recessive. And then freckles. Freckles on the face are dominant, lack of freckles is recessive. Right? There's more. There's like widow's peak and those kinds of things. So while we have some Mendelian traits, right, and we've been tracking them, most are polygenic. Polygenic traits are controlled by alleles at multiple genetic loci. Often, many genes are involved, and the phenotypic expression is quite complex. We see it more of going on a continuum of expression rather than yes or no, right? So some polygenic traits are things like your height, your skin color, your hair color, your eye color, your hair texture, right? Those are all polygenic traits. Okay? So here we have an example of a hitchhiker's thumb, right? It's one of the few Mendelian traits in humans, right? We have a hitchhiker's thumb on the left, right here, with a non-hitchhiker's thumb on the right. Next, we have sex-linked traits. These are traits that are determined by genes on the sex chromosomes, right? We call them sex-linked traits for that very reason. They have unusual patterns of inheritance and physical expression. As we learned in the last lab in Chapter 2, the X and Y chromosomes have key differences in the genes that they each carry. The Y chromosome has a limited number of genes, and they're largely tied to traits related to maleness. The X chromosome has many more genes that are important for both males and females. In XX females, 2 copies of the X chromosomes are present, so X-linked traits follow the same patterns described for Mendelian traits. In XY males, there's only 1 copy of the X chromosome present, and this may cause an unusual pattern of phenotypic expression. In most Mendelian traits, an individual has 2 copies of the gene, and this allows then for the homozygous and heterozygous genotypes that we previously discussed. However, an XY male who has an unusual X-linked trait on his X chromosome will have no alternative allele on a second X chromosome to compensate for it, right? So he doesn't have a second X chromosome to compensate for that unusual X-linked trait. Therefore, whatever allele appears at the genetic level on the X chromosome will appear on the phenotypic level in the individual's body. So whatever he has genetically, right, his genotype, is going to be expressed physically in his phenotype. In contrast, an XX female with an unusual allele on one X still has another X, on one X has another allele on her other X to make up for it, right? So if there is an unusual allele on the first X, then she still has a second allele that can have a normal allele so that it can counteract that unusual allele. The unusual trait variant then is less likely to be expressed in her than in an XY male with the same allele. The X chromosome has many genes, so there are numerous traits that are X-linked. A classic example is color vision. So humans, like many of our close primate relatives, have tricolor vision that is determined by 3 separate genes on the X chromosome. If an XY male has an alternative form of one of these genes, it may affect his ability to perceive certain colors. This results in a condition known as color blindness. It is estimated that approximately 5 to 8% of males worldwide have the most common form of color blindness, which is red-green color blindness. A male that has a color blindness allele may then pass it on to his female offspring, who inherit his X chromosome. However, his male offspring inherit his Y chromosome, so he cannot pass the allele to them. The expression and inheritance of the trait in females is a bit different. If an XX female has an alternative form of one of the color vision genes, she may still have full color vision because her other X will carry a normal version of the gene that makes up for it. These individuals are known as carriers for the trait because they carry the allele and may pass it on should the X chromosome be inherited by offspring, but they don't express the trait themselves. They might actually be unaware that they have that trait. It's possible for an XX female to be colorblind, but this is very rare. They would need the same unusual allele on both of their X chromosomes to manifest colorblindness at the phenotypic level. For this reason, X-linked traits are more likely to be expressed in males, but both males and females may have alleles for those traits and may pass them on to their offspring. So here's a look at the X chromosome and locations where we can see X-linked traits that can cause abnormalities. Color vision is down here, right, in this last section. And there's more, right? You can pause the video. This is also in your lab book, I believe, as well as your lab notes. I've put them in there so you can take a look at this and try it out, or just really look at the different X-linked traits that we have. Right. So for the most part, we have focused on Mendelian traits so far with one dominant allele and one recessive allele. Right. But Mendelian traits often have more complex dominance relationships between the alleles involved. The classic example in humans is the ABO blood group system. So I'm going to talk about it a little bit here. Just know the ABO blood group system is going to come up again when we talk about human variation. Different types of cell surface markers known as antigens are found on the surfaces of our red blood cells. In general, an antigen is a substance, such as a bacterium or enzyme or fragments of them, that triggers an immune response. Blood group antigens are either sugars or proteins. So the antigens expressed on a red blood cell determine the individual's blood type. And there are 2 main blood groups, ABO and RH. So ABO would give us blood types of A, B, AB, or O. And then with the RH, we have either RH positive and RH negative. So in the ABO blood group system, there are 2 antigens, antigen A and antigen B. The alleles for these antigens are found at one genetic locus, which means that ABO blood type is a Mendelian trait. But a person can inherit the A antigen allele, the B antigen allele, or the O allele. This is expressed, the O allele is just expressed as having neither antigen on the blood cell surface. So these alleles make 6 genotypes for the ABO blood system. So you have AA, which is A antigen allele inherited from both parents, AO, which is A antigen allele inherited from one parent and the O allele from the other parent, BB, which is B antigen allele from both parents, BO, which is B from one parent, O from the other, AB, which means A from one parent, B from the other, and then OO, which is the O allele inherited from both parents. What this comes with, or what this results in, is 4 corresponding phenotypes, right? So we have 6 genotypes, but 4 phenotypes, right? AA and AO genotypes have A antigens on the red blood cell, and it gives them the blood type A as their phenotype. BB and BO genotypes have the B antigen on the red blood cell, giving them blood type B as their phenotype. AB genotypes have both the A antigens and the B antigens on their red blood cells, which then gives them the blood type of AB. It's their phenotype. And the last one is OO genotypes, and they don't have either of the antigens on their red blood cells. It gives them the blood type O as their phenotype. So this discrepancy in the number of possible genotypes and phenotypes suggests that the ABO blood group system has a complex pattern of dominance and recessiveness. The A antigen and B antigen allele are dominant over the O allele. The only individuals with a true O blood type are homozygous for the O allele. This further suggests then that the O allele is recessive. The relationship between antigen A alleles and B antigen alleles is particularly interesting. The A antigen allele and the B antigen allele are co-dominant. This is when two alleles appear together as in AB individuals and both alleles are expressed in the phenotype. One is not dominant over the other, they are equally dominant. Therefore, when documenting genotypes in codominant traits like human ABO blood types, letters for the alleles are used, big A and big B, rather than the same letter in uppercase and lowercase, big A and little a. While much of the function of these red blood cell antigens is unknown, we do know that exposure to foreign blood antigens triggers a dangerous response from the immune system that can be fatal. The body will differentiate between its normal antigens, the self-antigens, and foreign antigens. Many foreign antigens, such as bacteria, trigger a protective reaction from the immune system. The body produces antibodies, which are special proteins that then attack the antigens directly or mark them for attack by other parts of the immune system. And then this process allows for the body to neutralize and fight off infections. So it triggers an immune response. In the case of blood cell surface antigens, the body will identify any foreign blood cell surface antigen and any foreign blood antigens and then produce antibodies to combat them. The antibodies will then attach to the antigens and cause the blood cells to then clump together. The coagulated blood does not travel smoothly through the blood vessels and they cannot get the blood and oxygen it needs. This condition, if not treated, can result in death. So because of this, it is important to know a person's blood genotype and phenotype to choose compatible blood for transfusions. Those with AB blood are lucky. They have both A and B antigens, which means they can accept A blood, B blood, AB blood, or O blood, right? Those with O blood, they have no antigens on the red blood cell, which means they can only accept O-type blood. On top of that, because they have O-type blood, they can give their blood to absolutely anybody. O blood can go to anybody because there are no antigens, so the body won't trigger an immune response to that blood cell, right? So it's very, very interesting to look at blood and blood groups and how they work. Right, you add in the RH factor, that's a whole another factor of positive versus negative, right? And you can look at motherhood and pregnancy and how the RH factor can work in that. It's very, very fascinating to look at these, at blood and the blood group system. But that's it for this chapter, right? Chapter 3. Next, we'll be looking at chapter 4. We'll be looking at forces of evolution. Again, there are videos, supplemental videos for this chapter that I have included in this module for this week.